Welcome to The Explainer. I am absolutely thrilled to have you here today. We're about to embark on a truly microscopic journey into, well, literally the most fundamental, magnificent architecture in the known universe, the cell. We're going to strip away all the complexities of the macroscopic world and zoom all the way down to the foundational building blocks that make every single living, breathing, growing thing possible. So buckle up because we are exploring the ultimate unit of life. Here's our quick syllabus for today. First, the fundamentals of life. Then, exploring prokaryotic cells. Third, eukaryotic cell boundaries. 4, the cell transport system. 5, the endomembrane system. And finally, number 6, the cell's powerhouse. Section 1, the fundamentals of life. Okay, let's dive into this. It's honestly incredible that whether you're a microscopic amoeba just navigating a tiny drop of pond water or a giant human composed of trillions of interacting parts, your basic structural and functional unit is exactly the same: the cell. The discovery of this cell theory is such a brilliant story of scientific progression. Back in 1665, Robert Hooke spotted these little hollow cavities in a slice of cork and called them cells. Fast forward a bit to 1838 and 1839, and scientists Schleiden and Schwann realized, wait a minute, all plant and animal tissues are actually made of these cells. But the big question was, where do they come from? Well, in 1855, Rudolf Virchow finally cracked the code, realizing that new cells are produced strictly from pre-existing ones. Now, as we look closer at these fundamental units, we hit a massive, and I mean massive, evolutionary divide. All cells are basically categorized into two main groups based on their nucleus. On one side, we have prokaryotic cells. Think of these guys as the minimalist survivors. They completely lack a membrane-bound nucleus to house their genetic material. And then on the other side, we've got eukaryotic cells, the complex architects. Eukaryotes boast a highly distinct membrane-bound nucleus to protect their DNA, right alongside a whole suite of other specialized membrane-bound organelles. What's also just astounding about cells is the sheer variation in their size. 0.3 micrometers. That's the length of mycoplasmas, the absolute smallest known cells on Earth. To put that extreme tininess into perspective for you, a typical bacteria might be 3 to 5 micrometers long, and a human red blood cell is way larger, coming in at about 7 micrometers in diameter. So yeah, life scales dramatically, even down at the microscopic level. Moving on to Section 2, exploring prokaryotic cells. Now, what's really interesting about this slide is looking at a typical bacterial cell's efficiency. Right in the center, you'll see that genetic material isn't locked away in some vault. It's naked, circular DNA just floating right there in the cytoplasm. Surrounding the cell is a super robust, 3-layered protective envelope. You've got an outer, sticky sugar coating called the glycocalyx, then a cell wall, and finally the plasma membrane. You might also spot some flagella used for movement or small surface bristles like pili and fimbria that they use for attaching to surfaces. It is an entirely self-contained survival machine. But beyond that main genomic DNA, a lot of bacteria have a secret weapon: plasmids. These are extra, small, circular pieces of DNA. Plasmids have a massive real-world impact because they bestow unique traits onto the bacteria, most notably antibiotic resistance. Yeah, this extra DNA is what allows bacteria to survive chemical attacks in really extreme conditions. Plus, in the lab, scientists actually use plasmid DNA to track the transformation of bacteria when they're introducing foreign DNA. They are literally nature's own USB flash drives. We can actually categorize these bacteria based on differences in those protective envelopes we just talked about. Specifically, how they respond to a lab technique called gram staining. Gram-positive bacteria, like Streptococcus, take up the gram stain because of their specific envelope structure. Conversely, gram-negative bacteria like E. coli or Salmonella do not take up the stain. This simple, color-coded test reveals some pretty profound differences in the chemical makeup of their cell walls. Section 3, Eukaryotic cell boundaries. You can think of a complex eukaryotic cell as a bustling, highly organized city. And any great city needs a secure wall to regulate trade, right? That's the plasma membrane. While the exact ratios vary depending on the cell type, lipids and proteins make up the vast majority of this wall. Back in 1972, scientists Singer and Nicholson proposed the widely accepted fluid mosaic model to describe this whole thing. Imagine a quasi-fluid sea of lipids, specifically phospholipids, with water-loving heads and water-fearing tails that gives the membrane a flexible fluid framework. Floating within this lipid sea are the proteins, acting as specialized transport gates, alongside carbohydrates attached to the surface. It's a dynamic, ever-shifting mosaic. Now, if you happen to be a plant cell, a fungal cell, or a protist, you actually get an extra layer of armor on top of that membrane, the cell wall. This is a rigid, non-living protective layer. In plants, it's mostly composed of cellulose and pectin. Unlike animal cells, which only have the flexible plasma membrane, plant cells absolutely need this rigid armor to maintain their shape, protect against mechanical injury, and survive harsh environmental variations. Mainly because, well, they can't simply get up and walk away from danger.
Section 4. The Cell Transport System.
So, our complex cellular city needs a highly regulated way to import essential supplies and export waste across its boundary. Understanding how things move really boils down to energy. Passive transport requires no energy at all. Molecules like neutral solutes or water simply drift along their concentration gradient, flowing from areas of high concentration to low concentration. It's a natural, albeit kind of slow, diffusion. Active transport, however, is an entirely different energetic narrative. So the crucial point is, active transport requires the cell to spend its own energy currency, ATP, to pump materials against the grain, from low to high concentration. It's rapid, it's highly controlled, and it's absolutely vital for large or polar molecules that just can't slip through that lipid bilayer unassisted. Section 5. The endomembrane system. Let's move to and see how this builds. Once supplies get inside the cell, they need to be processed by a highly efficient manufacturing plant. Step 1 is the factory floor, the endoplasmic reticulum, or ER. If it's studded with little ribosomes, it synthesizes proteins. If it's smooth, it synthesizes lipids. Step 2, products are sent to the shipping center, the Golgi body, to be packaged and modified, like, adding carbohydrates to make glycoproteins. Step 3, the finished vesicles are distributed to become storage vacuoles or specialized lysosomes for internal digestion. It is a perfectly synchronized assembly line. I want to pause specifically on lysosomes for a second because they are so fascinating. Discovered by Christian de Duve, these are often nicknamed the suicidal bags of the cell. They're basically isolated vesicles packed tightly with hydrolytic enzymes, which are essentially powerful water-based chemical scissors that operate perfectly at a highly acidic pH. Their main job? Destructive power. They act as the cell's waste disposal system, breaking down carbohydrates, proteins, and lipids. They even perform this wild process called autophagy, where they digest the cell's own old or damaged organelles to keep the cellular city clean and functional. Section 6. The cell's powerhouse. Okay, so all of the cellular manufacturing, active transport, and complex internal structuring requires an immense amount of energy. And that comes directly from the mitochondria. These structures are incredibly unique. They are double membrane bound, with the inner membrane folding into structures called cristae to greatly expand the surface area. This expanded area holds the oxysomes, which are tiny ATP synthesizing machines. But here is the most astonishing part for sure. Mitochondria are semi-autonomous. They contain their own circular DNA, entirely separate from the nucleus. They possess independent 70s ribosomes, the exact same kind found in bacteria, by the way. And they even divide independently via fission. Which leaves us with this final, truly provocative thought to end our explainer on. If mitochondria have their own unique DNA, and if they replicate entirely on their own schedule inside our cells, are we, in a way, just massive walking host cities for these microscopic ancient power plants? I mean, every thought you have, every single movement you make is powered by these tiny semi-autonomous entities living inside you. It truly makes you marvel at the profound complexity embedded within the single unit of life. Thank you so much for joining me on this microscopic journey.